So this is a course, the overall title is Cognitive Science in the Age of Artificial General Intelligence. My name is Alex Petrov. This is the second of 4 lectures. Yesterday we started, I introduced the motif that the overall kind of motivating background is that artificial intelligence is making these enormous strides, and that poses some very serious and important questions to humanity as a whole, and that cognitive science has a lot to contribute to this important conversation. And the first lecture was titled Layered Organization of the Brain and Mind. We had time only to go about halfway through it. So we dealt with some robots. So these are slides that we talked about last time. And the main idea of that was that it is not easy to survive in the world as an autonomous agent. There are these various things that need to happen. On the one hand, you have to be very reactive and quickly take care of some very rapidly moving objects. Gravity threatens to break you if you are not agile enough. And so this requires a certain kind of organization of the controller for your body. And this is the reactive end of the robotic spectrum. On the other hand, there are these tasks that involve remembering the past and imagining the future and planning ahead and making a plan about what will happen a month from today. And this is the deliberative pole of the robot control spectrum. And then a successful agent needs both. And that leads to this layered organization in which there are these bottom layers that take care of millisecond by millisecond muscle control. And then there are these top layers that take care about long-term planning. So this was the robotic side of things. And it turns out it is very closely parallel, and this is not accidental, the argument goes, to the organization of mammalian brains, because after all, mammalian brains are controllers of mammalian bodies, and mammals and all biological organisms are autonomous agents. And we talked about that brains have evolved from the inside out. There's this hierarchical organization of the brain. I took pains to emphasize that the subcortex is a very important part of the brain, arguably at least as important and maybe even more important than the cortex that everybody is somehow drawn to. This is about how far we managed to get, if I'm not mistaken. So there is a lot of neuroanatomy going on here. We're not going to try to define this in a systematic way, because that in itself is will take half of the available time. So those of you who know neuroscience are in familiar territory. Those of you who don't know neuroscience, just kind of let the terminology wash over you and don't worry about too much which brain structure we're talking about. I know it can be overwhelming. I was, and I'm still kind of learning this as we go, but the slides I've taken some care, and it took me a lot of time to make them anatomically accurate, but the big principles are what I'm getting at here. And to get to the big principles, you don't need to know the exact anatomical locations of things. The big principle is that the brain has evolved from the inside out. The deepest, most central structures are the evolutionarily oldest and the functionally most important. And with robots, we can kind of construct them one layer at a time and see what happens. With biological organisms, evolution has already done the construction for us. And so to understand the layered organization, one approach is to take pieces of the brain out. So there is a brain with many layers. We cut some of the layers and retain some of the other layers and see what happens. And that program has been carried out, this research program has been carried out a long time ago, 50, 60, 70 years ago. So I will be reporting some quite old data. And as I was going over, I have a more general comment. As I was going over my lectures for today, I realized maybe 2/3 of the material dates back from the 1950s and 60s. So there are these data that everybody should know about, but somehow they don't feature very prominently in textbooks, at least not in the textbooks that I have come across. And I have been going out over many textbooks, believe me. So there is a need to kind of integrate more of the existing knowledge base of our science. So then there are these studies involving animals with damaged brains, and this table is taken from a textbook on neuropsychology by Kolb and Wishaw. As you'll see from the references on the subsequent slide, these were some of the actual researchers who were doing this many years ago. So there are these different pieces of brain, and there are these different terminology about different ways of experimenting, kind of cutting the nervous system at a given point. And depending on where you cut and what the name of the animal, let's say a spinal animal is an animal that only has a spinal cord and nothing else. And then you have a low decerebrate, which has a spinal cord plus the hindbrain. Then you have a high decerebrate, which has a spinal cord, hindbrain, and midbrain. And then you have diencephalic, which has spinal cord, hindbrain, midbrain, and interbrain or diencephalon, and so forth. And so let's go over some of these. This is the overall plan. So I will be referring to these terms. I hope, I realize it is kind of small font, but I hope you can still see. So hindbrain is the thing that connects to the spinal cord. We're not going to deal with the spinal cord, although it's obviously important too. So taking the spinal cord for granted, the first thing actually inside the skull is the brainstem, and that is part of what the anatomy is called hindbrain. And then on top of that, also part of the hindbrain is the cerebellum, which is very important for motor control. This is another view of the cerebellum in sagittal cross section. That is a section down the head. If you cut the head down the middle like this and then open the two halves like a book, that would be a sagittal cross section here. So this is the brainstem kind of split down the middle. This structure, medulla oblongata, the name in Latin means zucchini, I think, and because it has this shape, so imagine a zucchini, cut it down the middle, open it up. All right, so then that's the lowest level that we're going to deal with, and then I will introduce the higher levels as we proceed. So an animal that only has, first of all, this reminds me, I should mention something. A person with a spinal injury, that is to say, if you take an intact human being, and they are people, the unfortunate people like that, they break their neck and they disconnect the spinal cord from the rest of the brain, and that makes them paralyzed from the neck down. A very unfortunate situation, as you can imagine, but you're still yourself, you're fully awake. Yes, you're paralyzed, but you have emotions, almost not 100%, but 90% of emotional life is preserved. You have your thoughts, you have conscious, and so forth. And so that disconnecting the spine from the brain doesn't undermine the working of the brain. It just abolishes the connection of the brain to the body. And that's a big problem, but that's a separate, it's a kind of communication issue. Now, by contrast, if you damage the very low levels here of the brainstem, there are centers that are whose damage is incompatible with life because they participate in the regulation of the vital functions of the body. Things like breathing and heartbeat and all kinds of vital functions. So many of these individuals simply die. And then the other thing, at the very low levels of the brainstem, controls consciousness in the sense of like conscious versus unconscious. And so low decerebrate animals have difficulty maintaining consciousness. So they just lie there unconscious. So this is the lowest level. If you damage that, it's just you're dead basically. Even if you maintain breathing, or you can be kept alive using a breathing machine from the outside. So that's not of interest to us. Our interest begins with patients that have damage a little higher up. I should probably be saving time, so I will just very briefly mention damage a little higher up. You may kind of come into consciousness and out of consciousness, and this is called persistent vegetative state. And this picture here is a famous patient by the name of Terry Schiavo. It was all over the news about 20 years ago. There was this big controversy, should her life support be continued or discontinued? And the parents said yes, and the husband said no, and who decides? And the court in the end had to decide, and it was a big drama. So there are very many ethical issues involved in this medical research and things like that. But let's move on in the interest of time. The reason I'm putting this into my slides is so that I can tell you about this point onward. The other ones are kind of foundational, but kind of medical. They don't have to do with control of behavior. They have to do with keeping the body alive, basically. So control of behavior begins somewhere here at the level of what in this field. The field in question is neuropsychology, the behavioral deficits following brain damage. This is the subject matter of the science of neuropsychology. So neuropsychological term is high decerebration, and that involves cutting the brain along the line here. There are many different versions of this, but for our kind of high level overview, let's imagine there is a line separating the diencephalon from the mesencephalon on the other. So you have an intact spinal cord, you have an intact brain stem. Also, all of these structures here, brainstem, pons, tectum, tegmentum, cerebellum. In the tectum, you have the superior and inferior colliculi, which contain inputs from the eyes and the ears. So these animals have some visual inputs and some auditory inputs, so they can see and they can hear up to a point. And then in the tegmental area here at the bottom, there are a number of motor nuclei and some very important additional structures, notably the so-called ventral tegmental area, which is a major source of dopamine in the brain, and the so-called periaqueductal gray. This is a typo, it should be PAG. Okay, so there are certainly very important, basic, ancient structures of the brain that are preserved. And now, look what happens. These animals can respond to distant objects by moving toward them. So first of all, they can move. They don't just lie there. They can move. They can walk, stand, resume upright posture when turned on their backs. Remember the robots? There was this robot that slipped on the ice and was kind of standing back up. These animals can sort of do that, although with impairments. A lot of that is actually the job of the cerebellum. And by the way, I'm not going to tell you much about the cerebellum or the little brain, but it is super important for motor control. It contains the majority of the neurons in the brain, it turns out. If I remember correctly, 60% or 70% of the neurons inside your skull are actually located in the cerebellum. There are more neurons in the cerebellum than the rest of the brain. So, something to think about. So then, high decerebrate animals can respond to distant objects by moving toward them. They can walk, stand, resume upright posture when turned on their back, and even run and climb when stimulated. Thus, all subsets of voluntary movements are present at the subcortical level of the midbrain. And one analogy that comes to mind, kind of it involves certain idealization and simplifications, but it's still useful, I think. It is like, think of the piano and the keys of the piano and the different kind of movements, elementary movements, like standing up or making a step forward with your left leg. These are like the notes of the piano, and there are these different keys. And then you can play different melodies by pressing on these keys in different sequences. And so the midbrain has all of these elementary components, and then the higher levels of the structure play them out in different sequences. And there is a lot of analogy here with, if I go back to my robotics section, all the way back to Shaky the robot. Remember these things here? So low level actions and intermediate level actions. So these elementary sequences are the low level actions of the mammalian brain, and they reside in the in the brainstem and the thalamus, not the thalamus, sorry, the superior colliculus and the tegmentum and the cerebellum. So over up to here. Okay. High decerebrate animals can also effectively perform automatic movements, not just elementary piecemeal movements, but integrated sequences, such as grooming, chewing food, rejecting food. So if you put some kind of rotten meat in the rat's brain, it will spit it out. If there is bitter water, they will kind of chew out, spit out the water, things like that. This is rejecting food. And these are instinctual movements. Newborn infants already have them. And here are some examples in adult humans. We can all recognize these things. They're very ancient kind of mammalian patterns of eating and drinking and so forth. Okay, so moving on. Oh, yes. And most of this research has been done with cats and rats and dogs, but there are some unfortunate human infants who are born without a forebrain. In other words, they are high decerebrate. Due to developmental abnormalities, they're just born and half of their head is missing. This is a MRI image of one unfortunate child like that. It's a 3-year-old mesencephalic, so the mesencephalon, the midbrain, is more or less preserved, but the higher structures are missing. They show many behaviors of newborn infants, periodically sleeping and waking, sucking, yawning, stretching, crying, and following visual stimuli with the eyes. So if you put a toy in front of their eyes and move it left and right, their eyeballs kind of track the motion. Even though they can sit up, they show little spontaneous activity, and if left alone, remain mostly in a drowsy state. But the startle reflex is there, so if there is a loud noise, they will kind of startle and turn, things like that, in the same way as typical infants do. Okay, so moving up the scale here. So if we add one more level, diencephalon, also known as the interbrain, it has several important structures, notably the thalamus. Thalamus in its own right is like a little brain of its own. It has a lot of subdivisions within it. For example, Those of you who study vision, you have heard a lot about the so-called LGN, lateral geniculate nucleus. This is the first relay station from the optic chiasm into the brain. And so LGN projects to the primary visual cortex and so forth. So the thalamus has a lot of sensory nuclei, a lot of motor nuclei, and there is a lot of thalamic areas that are in close bidirectional connectivity to neocortex. In many ways, the thalamus is like the 0th level of cortex. As you know, cortex has 6 layers. There are these cortical layers, and then thalamus is usually kind of point-to-point connected. So if you don't have neocortex, the thalamus is like the first stage into the development of that. So thalamus has better vision compared to the superior colliculus. It has better audition, better sound processing compared to inferior colliculus. Also, these animals have intact olfactory system, which for things like rats and dogs and cats is very important. This is their primary sense. And very importantly, so that's the thalamus. Hypothalamus, a separate structure located under the thalamus, so hence the name, and the pituitary gland. This is the interface between the brain and the hormonal system of the body. So the pituitary gland is sometimes labeled the master endocrine center. So it releases these hormones that regulate other hormones in turn. And so hypothalamus is a super important part of the brain. We'll meet it later when we talk about what happens when you electrically stimulate the hypothalamus. Very interesting things happen. So it is involved in homeostasis, that is maintaining the balance in the body. Things like thermoregulation and hunger and thirst, they all have representations in the hypothalamus. So this is what these animals have. What is missing is the so-called telencephalon. And so the basal ganglia are missing and the neocortex is missing. And also the hippocampus, amygdala, all of these things are missing. But you have spinal cord, brainstem, thalamus, hypothalamus, and substantia nigra. Substantia nigra is another major source of dopamine in the brain. So when you have all that, what can you do? So these are the so-called diencephalic animals. The hypothalamus integrates the body's physiology with the brain's activity. So now the brain is kind of receives signals from the visceral body. So it knows about with hunger, thirst, thermoregulation. And so diencephalic animals can thermoregulate. What does that mean? If they are cold, they start shivering, thereby generating heat. If they are warm, they start panting, therefore kind of evaporating some moisture from their tongues, and that cools them down, things like that. This is active thermoregulation. The animals from before that we talked about without the hypothalamus, they don't shiver, they don't pant, they don't thermoregulate. So these animals, the diencephalic animals, thermoregulate successfully, but they do not eat, drink well enough to sustain themselves. So you have to feed them by kind of injecting, not injecting, but putting food in their mouth, and then they kind of chew on the food. But they cannot kind of take the food out of the plate themselves. Now, very important, if we talk about emotions, which is what we're getting towards, diencephalon is like the beginning of emotional life. It adds affective and motivational dimensions to behavior in the sense that it becomes energized and sustained. So remember, this mesencephalic child remained mostly in a drowsy state. So without the diencephalon, you just sit there doing nothing. But once you have these next level up, motivational, emotional centers, then you stand up and start exploring, moving around, And sometimes even getting these exaggerated emotional states, like well, there is this term sham rage. So there are like a cat will all of a sudden get angry and start attacking the air and exhibiting the kind of stereotypical aggressive behaviors that are species appropriate for cats. For example, lashing the tail, arching the trunk, making leaf movements like this, hissing, and you know cats, or at least I've grown up with cats, and I anyway. Okay. Also, so these are kind of an external manifestation. The skeletal musculature, it moves in an appropriate pattern, and also sympathetic signs of rage. That is the reference to a sympathetic nervous system. This is an autonomic nervous system. It controls things like perspiration, pupil dilation, urination, high blood pressure, high heart rate, increases in certain hormones, stress hormones, such as epinephrine and blood sugar. And without a kind of moderating and inhibitory control of the higher levels, these emotional attacks sometimes last for hours. So the cat could be fighting the air, let's say for 30 minutes, and then falling asleep of exhaustion. And similar emotional attacks also occur in people with hypothalamic lesions. And one general kind of theme is humans are mammals, and so we make no exceptions to cats and dogs, especially as far as the low kind of subcortical levels are concerned. The thing that sets us apart is our very developed neocortex, but the subcortical levels are very much conserved from our cousins, the primates and mammals more generally. Moving on, we get now to the so-called decortication procedure, which is to leave the brain everything except the neocortex itself. And so the new structure that you are adding to the mix before is a very important structure called the basal ganglia. So let me tell you a little bit about the basal ganglia, so kind of crash course in anatomy. So this is a human brain, a kind of semi-transparent brain. So of the brownish thing is the cortex. These purplish structure are subcortical structures, are various kind. And so there is this collection of big subcortical inhibitory nuclei. A nucleus is a kind of place in the brain where neurons live. I mean, the bodies of the cells are actually located in the nucleus. So a kind of clusters of neurons, or nuclei for short, they have different names. So the basal ganglia, there are several of them. Striatum, which in turn is divided into putamen and caudate nucleus. They have this funny shape. Caudate in Latin means tail, so tail-shaped nucleus. Another structure, globulus pallidus, and amygdala, very important for emotion, especially fear processing, is also classified as one of the basal ganglia. So these things are located above the thalamus. So these egg-shaped things are the thalamus. So the thalamus sits on top of the brainstem. There are two thalami. Everything is kind of left and right. And then on top of that, and as I said, the thalamus is very tightly integrated with neocortex. So these white matter here, these are nerve fibers covered in this fatty insulation called myelin. That gives them their white color. So the thalamus is connected to the cortex. I assume you can see my mouse. Right, Jenny? Okay. So, and then all of these cables, they have to go through the basal ganglia. And so here in the upper left-hand corner, you see another diagram. The fibers of the white matter kind of go through the basal ganglia, and the basal ganglia have these holes to let these cables go through. A very interesting organization. You can kind of tell the thalamus was there and the basal ganglia were all there was, and then the cortex evolved and it connected to the thalamus and it had to kind of negotiate the connectivity of the system had to kind of go through the pre-existing structures. Okay, so we need to keep going. I don't want to run out of time too badly this time. Now, what can a decorticated rat do? And now here is where it gets mind-boggling. I remember when I was first reading this 25 years ago, I said, wow, who would have thought? So with a little training, decorticated rats find water and become able to maintain themselves on water and laboratory chow. So they can kind of, when they're hungry, they go and eat. When they're thirsty, they go and drink. You don't have to give them things in their mouth. They take care of themselves. They have normal sleeping, waking cycles, run, climb, and swim, even negotiate simple mazes. They can also sequence a series of movements, such as copulation or grooming. So the rats, when they groom, they kind of clean up their whiskers with their paws, and they kind of go over behind their ears and so forth. So a complex kind of species appropriate sequence of skilled movements. They cannot do all of that. To a casual observer, the decorted rat appears indistinguishable from normal rats. And as a matter of fact, and I read these books about effective neuroscience, and they say a standard kind of exercise for neuroscience students is to challenge them to tell which group of rats are normal rats with intact brains and which groups are decorticated rats. And more students think the decorticated rats are the normal rats than vice versa. So people are below chance in telling them apart. And the reason for that, I'm told, is that decorticated rats are more active and they kind of run around more and so forth. Okay. So the decorticated rat does indeed have a lot of behavioral difficulties, but seeing these difficulties, deficits, requires a trained eye. So you have to know what you're looking for. All the elementary movements that animals might make are part of the behavioral repertoire after decortication. As I said, they can walk, eat, drink, and so forth, and even raise litters of pup in a seemingly adequate fashion. Now, what about humans? It turns out there is a condition which basically amounts to decortication. There is this terrible disease called hydranencephalia or hydranencephaly. I'm not sure I pronounced this right, but, and by the way, it is not to be confused with a far more benign condition called hydrocephalus. So hydrocephalic kids are a lot better, in much better shape compared to these that we're talking about here. So typically this condition, hydranencephaly, is caused by a brain stroke in utero, inside the mother's womb. Some unfortunate embryos suffer a brain stroke, a kind of interruption of the blood supply to their brain before they're born. And so a lot of the brain dies, and there are certain arteries that feed the cortex, and when these arteries are blocked, the cortex dies, and the skull kind of develops normally, more or less, but the missing cortex, the volume of the cortex is now full of cerebrospinal fluid with salt water. And hence the name hydranencephaly is there is no encephalon, and anencephaly is the lack of cortex, and the hydro is that there is a lot of water involved. So this is a scan of one of these children. You can see some remnants of the occipital lobe here of the cortex, but typically, even though they're kind of there, they're not hooked up properly to the rest of the brain. There is no white matter and so forth, and so they're not functional. Even though some remnants are there, the cortex is basically all gone, but the basal ganglia are intact. And now, what does the important thing on neuropsychologists would ask is, what are the behavioral signatures of this? And now, very importantly, this condition sometimes is not diagnosed until several months postnatally. So their parents, they think they have a normal infant for several months. Just like to a casual observer, the decorticated rat appears indistinguishable from normal rats. To an unsuspecting parents, this poor child who had a brain stroke before it was even born and the cortex resorbed and turned into water, the parents don't know for several months after birth. And the reason for that is that even normally developing children, they have a lot, their cortex is very much underdeveloped at birth. So we are born prematurely, basically. Our brain is not fully formed at birth, and it continues to develop for many months after birth. And so these children are awake and often alert. They show situationally appropriate emotional or orienting reactions to environmental events. Although, and this is now the difference, I had a table somewhere with some numbers. If we go back here, yes. So cortex is 31% of the volume of the brain in a rat. It is 77% of the volume in a human. And so when the rat loses 30% of its brain, the remaining 70% can still keep the rat in such a shape that an untrained observer cannot tell the decorticated rat apart from normal rats. For a human, the cortex is like 3/4 of the brain. And now if you lose all of that, you are no longer indistinguishable. You are severely impaired. So yes, they are indistinguishable soon after birth. Where am I? Here. But after age 2 or 3, they start missing developmental milestones, and then it becomes very apparent. Cortex is very important for human beings. We are not humans. We're not fully human without neocortex. So these kids, they cannot really walk. They cannot really move about. They lie there, but they can react. For example, the example I read, and this paper, by the way, is a great paper here, published in the journal BBS, Behavioral and Brain Sciences. I recommend it very much. So the example given involved social emotions. So this little girl had an even younger sibling, and when you put her sibling in her arms, she would kind of hug it gently and smile and things like that. So these are emotional reactions to the presence of her sibling, things like that. So very interesting. But I'm mindful of the time, so let's keep going. And that finally brings us to neocortex. As I was saying, so this is now an intact brain in a coronal section. Coronal section means if you cut my head like this, of tracing the outline of my headphones, and then take the front away and look at the remainder, this is what you will see. So this is the brainstem now cut the other way. This is left hemisphere, right hemisphere. This is the left thalamus, the right thalamus. These are the basal ganglia. And this is now the cortex, all the convolutions and so forth. And these are the connections connecting the thalamus to the cortex and back. And so the cortex is bidirectionally connected both to the basal ganglia and the thalamus. And the thalamus is bidirectionally connected to the basal ganglia. And so the three things form a very tightly integrated triangle called striato-thalamo-cortical loop. So striatum, remember, is one of the key components of the basal ganglia. Where is my basal ganglia diagram here? Yes, striatum is here. And so this caudate nucleus, all of that is striatum. And so striato-cortico-thalamic loop is basal ganglia, cortex, and thalamus. So to understand what the role of the cortex is, you have to ask yourself, what is it that decortical animals cannot do? And so what is it that they cannot do? And so decortical animals do not build nests, although they engage in some nest building behaviors. So they know how to move in order to build a nest, but they don't kind of plan ahead far enough that, okay, now it's the kind of nest building season. I better build my nest. They do not hoard food, although they might carry food around. So they can perform the motion. It just doesn't occur to them that's the thing to do. They have difficulty making skilled movements with tongue and limbs. Yes, so fine motor control depends on cortex. And this is why under stress, the kind of cortex is down-regulated. More of the control of behavior goes down to subcortical structures. And very frightened people, they lose kind of fine motor control. Socially anxious people, their voice trembles when they speak in public. There are all kinds of telltale signs of when the cortex is under stress. Now, decorticated animals can do pattern discriminations if the tasks are relatively simple. For example, a decorticate could discriminate two pure tones, but would be unable to distinguish complex sounds. And let's not read all of that. So in sum, the cortex does not seem to add much in the way of new movements to an animal's behavioral repertoire, and neither is it essential for learning. There are many experiments with decortical animals. They can learn Pavlovian conditioning, operant conditioning, all kinds of behavioral learning protocols are preserved at the level of the basal ganglia. So cortex is very much involved in learning, of course, but it is not needed. You can learn without the cortex. So what the cortex seems to be mostly about is that it extends the usefulness of all of these behaviors or make them adaptive in new situations. And so putting all of these things together, you see there is this kind of mammalian heritage. Our brain has these different pieces, and one can see them kind of coming online one by one, and each of them kind of builds on the pre-existing ones and enriches them and so forth. And of course, in an intact brain, all of these things talk to each other, and they higher structures kind of regulate the lower ones, but the lower ones also kind of affect the upper ones. And it's all one integrated system, which leads to this selection problem coming up. But before I get to that, I have this one more slide. It's a hypothetical example. This is not based on some kind of hard the program of research, just a kind of story that puts the things together in a reasonable way in an ecological context. So consider the mechanisms of defense, which is highly related to the emotion of fear. So if you have to run away from scary things and protect yourself from being eaten by predators, there are a lot of things at different levels. So for example, at the level of the spinal cord, you have this reflexive withdrawal. So if I put a kind of acid on your skin, you will flick it off, and that is a spinal reflex. Hindbrain is this startle responses. So if there is sudden noise behind you, you will turn and look, and we will see examples of that in a moment. Then species-specific responses, these freeze and fight and flight. So this is now not just startling, but kind of recognizing, oh, this is a snake. I better run away from the snake. And then these higher level thalamus, sensory cortex, and hippocampus and septum, and they, in this author had kind of put them, they all talk to the amygdala, and then the amygdala gets out. And then finally, cognitive analysis is about, oh my God, I have a lecture to give tomorrow and my PowerPoint slides aren't ready yet, so I better stay home late and work on my lecture for tomorrow, that would be all the way up here. So you see the different levels and how they kind of build on top of each other. Okay. Now, selection problem. We have all of this machinery. And what if the different pieces disagree and they give different recommendations? This is known as a selection problem. More generally, a selection problem arises whenever two or more competing systems seek simultaneous access to some limited resource. For example, each of these kittens wants to be king of the hill, and there's only one throne, only one of them can be on top, and so they fight for who will be on top. And so that gives you, coming back to our kind of overall arching theme here, evolution and all of that. So organism is an autonomous agent. What is the key limited resource? Maybe think about it for 5 seconds. I should give you time. So what do you think is the primary resource of a biological agent? Okay, unfortunately, we couldn't really, because it's not a real classroom, I cannot make it really interactive, which is a pity. But so the agent's body is the answer I have in mind. And it is limited in the sense that, yes, you have different limbs and you can move in different ways. But if you're running away, you're not hiding. And if you take the left turn in the maze, you're not taking the right turn in the maze. So you have only one body, and many modern movements require the engagement and balance of the entire body. You cannot kind of walk with your left leg and run with your right leg. You must walk with both legs in a coordinated way. And so this gives rise to this action selection problem. Here's a specific example. So suppose the overall conditions are such that there are these three behaviors that are being considered as relevant for the situation at hand. You're very hungry and there is food around, and so you can eat. But in order to eat, you have to move left. There is a room full of food. You have to go to the food feeding room and eat. Or you're also thirsty. You have to go to the well and get some water and drink. But that's a different place. You either go one or the other. And then overall, it's a kind of bad time of the day. There are birds flying overhead. And you may, if you're a little mouse hiding in the grass, you're maybe better off kind of hiding, keep hiding, because the predatory birds otherwise will spot you, and that's a problem. And so there are these three things, and somehow they compete, and all movements are inhibited until a decision is made. So there is no behavioral output. And then at some point, a decision is made, and now the animal is committed and it starts moving. And let's say in this particular example, it decides to go to the food source and start eating. And now there is this analogy involving a dam. The basal ganglia have this function, a very important, very prominent theory of the role of the basal ganglia in the mammalian brain is that they act as an action selector. And so they're inhibitory nuclei. So they inhibit everybody, and they selectively disinhibit one of the multiple options. So it's like having a car with 5 different engines, and you have 5 braking pedals, and all the pedals are always depressed. You're always slamming on the brakes on all 5 engines, and then you gently release the brake on one of the 5. This is disinhibition, selective disinhibition. And there is a prominent theory of the role of the basal ganglia in terms of disinhibition. There is this very interesting paper building a mathematical model of the basal ganglia, and there is this mathematical formulation of the action selection problem. The mathematical framework for that is called multi-hypothesis sequential probability ratio test. So that's a mouthful, but you accumulate evidence. You have these likelihood ratio tests. So the overall idea is just staying to the level of very high level intuitive principles. So you accumulate evidence, you estimate the so-called expected utility of each candidate action. So you consider multiple alternatives, you accumulate evidence about utilities of these different alternatives. Then there is this important step of converting all of these disparate measures into some kind of common scale. So you have to compare thirst against hunger, for example. So they must somehow decide, am I more thirsty or more hungry? Which of the two will take over in this particular case? And then there is some form of winner takes all selection. And so the basal ganglia perform these operations. So some structures calculate the utilities, convert them to common scale, and then the output nuclei of the basal ganglia take the maximum, disinhibit the action with maximum utility. And there is a nice mapping. This paper here, Lapore and Gurney, Neurocomputation 2012, make a nice kind of mapping between the equations of their mathematical model and the different structures in the basal ganglia. There is this very complicated anatomy. There is a direct pathway, an indirect pathway, a hyperdirect pathway. Those of you who know neuroscience, hopefully these terms sound familiar. So for our intended purposes, we don't have to worry about that. The take-home message is that, and that's the important thing. So let me slow down and emphasize it properly. So coming back here, the fundamental limited resource is the body. Competition about the body is resolved in the sense of who takes control of the movement of the body. The body has to move as one unit. If the right hand is doing one thing and the left hand is doing another thing, it becomes a mess. So the body must move as a unit. And in motion control, they have this very important concept, the final common motor path, which is the highest level of the system where different competing alternatives converge, and then only one is selected, and then from that point on, it is a single motor path going down. So here in this diagram, there are multiple alternatives being considered, and then they converge into some kind of decision maker, and then downwards, it's a single common path going down. And so in the human brain, mammalian brain more generally, the basal ganglia are the final common motor path, not the neocortex. And so the basal ganglia gate the motor system below them by inhibiting the thalamus. And also, interestingly, through the thalamocortical loop, they can also strongly influence what goes on in cortex. And I don't have time here, but it's a fascinating story. It's a whole story for another time. But the hypothesis is the basal ganglia evolved primarily in the service of motor control to solve the selection problem. And as you know, to open some parentheses, damage to the basal ganglia, such as in Parkinson's disease, have kind of lead to motor deficits. These people have trouble initiating movements and so forth. But they also have cognitive deficits, Parkinson patients. And so the hypothesis I was referring to is there are also cognitive tasks that involve selection. For example, working memory control. Should I commit this to memory or should I ignore it? There is a control problem involving gating memory, and the basal ganglia are already there, already doing action selection in the service of motor control. And so the hypothesis is they just took over additional functions in service of cognitive control. And so now they're doing both. They're doing motor control and also heavily involved in cognitive control. And that is interesting because they're evolutionarily older and kind of in terms of sophistication of the information processing going on there, they are less sophisticated compared to neocortex. Now, at this point, you can, if I wasn't speaking too fast, so fast, so you can, you're just struggling to kind of understand what I'm saying, and my Bulgarian accent only helps the Bulgarian people maybe, but non-Bulgarians probably struggle with my accent. Anyway, so if you had time to kind of slow down and think, we'll say, wait a minute, what about diencephalic animals? In other words, there were all of these guys that did not have basal ganglia at all, and they were not just sitting there doing nothing. They were running around, grooming, and so forth, having sham rage attacks. How does that happen? So if the basal ganglia does the action selection, if you take it out, why doesn't everybody becomes a mess? And that's a very good question. And there is a very interesting paper which puts, so by Prescott, 2007, they put up this interesting hypothesis. It is not, so the basal ganglia story is very well established. There are hundreds of papers, dozens of models. So this is very well researched area. Of course, there's a lot of left to learn, but at least the broad outlines of the story are in place about the basal ganglia. Now, this thing is a lot less developed. I only know about papers from this one author and his collaborators. But the idea is, below the basal ganglia, there is a brainstem substrate for selection. And that other selection center, brainstem, very early on. So this is the rat brain. Brainstem is here, the earliest part over the spinal cord. So even there is a kind of very preliminary action selection center in something called, in a structure called medial core of the brainstem reticular formation, some part of the brainstem. And by the way, the core means near the midline, and the midline is the evolutionarily oldest part. So the brain grows from the bottom to the top and also from the middle to the midline sideways. So midline structure, medial structures are ancient, dorsal structures and lateral structures are evolutionarily newer. So the very ancient part of the midbrain, of the brainstem, excuse me, the medial reticular formation seems to involve some kind of basic behavioral switching. And so it can maintain that while the higher up structures are still developing or when they're damaged. And so the mammalian brain appears to have two systems for action selection. One right on top of the spinal cord, and then another one here in the basal ganglia. And so now, if it's two of them, again, the question arises, what happens if they disagree? How are they coordinated? And so in this paper, there is some speculation, really. They say we don't have enough evidence. It's too early to tell. They consider two possibilities. I put the third one because it kind of integrates the other two. And on my kind of a priori belief is that the brain is complicated and everybody talks to everybody. And so anyway, so then one of the several possibilities is that the basal ganglia and the medial reticular formation are in this configuration here. So they both receive sensory inputs. So they must know what the state of the body is and what the state of the outside world is in order to make informed decisions. And the MRF is sitting on top of the spinal cord, and the spinal cord is connected to the muscles. And so the effectors are here on this diagram. But then there is another structure called, with some funny name, it doesn't really matter. There is a name in Latin that is abbreviated PPN. And so the basal ganglia inhibit that. They inhibit everybody, and they selectively disinhibit one of the many things they inhibit. So They inhibit this PPN, and then PPN is connected to MRF. So long story short, there is some circuit, and the two decision makers form these recurrent loops, and somehow in the intact overall animal, they have this coordinated response. Now, if you damage the system and cut the basal ganglia away, the MRF is the only one left standing, and it is still sufficient to provide some basic behavioral outputs. And so this diagram has aspects of layered control, or layered meaning the top structure inhibiting the bottom, and so the ultimate decision maker is the basal ganglia. That's the layered aspect. But the hierarchical, or sorry, this is the hierarchical aspect. The top kind of hierarchically imposes its will on the lower structures. The layered aspect is that the lower can still kind of function in the absence of the top, and also to some degree modulate what's going on upstairs. And now, the last time we asked ourselves, can a less intelligent entity control a significantly more intelligent one? And in some sense, the cortex is the most intelligent, at least the most sophisticated information processing in the human brain takes place in the cortex. But as you can see, there are all kinds of important machinery going underneath, and the final common motor path ends at the level of the basal ganglia, which is a subcortical structure. And I want to introduce another term because it's important. In the affective neuroscience literature, they sometimes talk about the so-called centrancephalic core, which is, if I go back to the diagram, the brainstem and the midbrain and the diencephalon. So this is the centrancephalic core. These are these decision-making, emotional, body regulating centers of the brain, and they're proposals that they play a key role in consciousness. They give you a sense of an integrated self. I'm this one agent. The I is generated there. Okay, so the brain centrancephalic core energizes the neocortex. So it controls sleep and wakefulness and attention and motivation. And also, the brain centrocephalic core performs action selection. The cortex proposes what to do, but the basal ganglia decide what to do. And now, coming back to my opening about artificial intelligence, there are people who say we humans will be, with respect to these super intelligent computers, in the same relation that the basal ganglia are with respect to the neocortex. Namely, we control wake and sleep cycles, so we turn them on and off. By the way, when you sleep, you're not really turned off. The brain is very active during sleep, but at least the behavioral outputs are inhibited, so you lie down and you don't move. We will be in control of computers, and we will be deciding what of the options that computers calculate for us to take. Now, that is the hope. There are reasons to be skeptical about that, because we don't inhabit the same skull, as I said last time. But I see that it is over an hour into my time for today, and I know Kalina Christoph is giving a lecture this afternoon, so we cannot go over time. So let me stop sharing this PowerPoint and move on and to this second lecture for the day, which is about unification of cognitive science and affective neuroscience. So just a moment, I want to make sure. Do you see my slides? Yes, no? Okay. Okay, now, emotions. It turns out they're super important, as we'll see. Last time, we introduced this example already. So there was this very famous and influential model of linguistic natural language processing 50 years ago. Just like the chatbots of today, you could ask it questions and it will answer. And if you keep asking a series of why questions, why did you do that? Why did you do that? Why did you do that? At some point, the model said, because you asked me to. So the final goal originates with the human operator, not the model. And it turns out, so biological organisms are not like that. They move by themselves on their own, kind of under their own steam. So they have a motivational system that makes them move in the absence of external prodding. And emotions and motivations are two sides of the same coin. So when I was thinking seriously about these problems having to do with superintelligence and the problem of control and the alignment problem, so what are the goals and all of that, and what are human goals? I realized that my cognitive science training was not sufficient, because the moment you start talking about goals, you talk about values and motivation. And the moment you start talking about that, you are in the realm of emotions. And so you have to talk about emotions. And that is why my appeal is that cognitive science should get out of its unfortunate state of relative neglect of emotion and start paying a lot of attention to emotional processing. And so I see I have 20 minutes. That should be just enough time to give some examples of emotional episodes. And again, as I mentioned before, there will be some experiments done in the 1950s, super informative experiments, so informative, in fact, that they're probably not going to be to pass IRB approval. They will be considered unethical today. And so we are kind of in this dilemma. The most informative manipulations tend to be kind of nasty and unpleasant. And so there is this trade-off. And in the 1950s, there were less regulations. And so some scientists were doing some experiments that today make us cringe. But the knowledge is very valuable. And so part of the suffering that was inflicted 70 years ago to some unfortunate monkeys, for example, pays off because we learn so much from that. So without further ado, let me get to what is described as perhaps one of the greatest neuroscientific discoveries of the 20th century, which is self-stimulation via implanted electrodes. So there is a way to insert a kind of needle-like electrode into the brain, into different parts of the brain. All of the electrodes I will be talking about today go to the subcortical structures, all the way down to the brainstem and to the diencephalon, the emotional circuits of the brain. And so you have this electrode, and in the 1950s, this technique was first developed. And this is known under the kind of the colloquial term wire heading, because there is this wire sticking out of the head. And so this is a wire-headed rat which has these electrodes inserted in certain parts of its brain. Specifically, the so-called medial forebrain bundle, a particular anatomic structure around this ancient medial line of the brainstem. I have a picture coming up later. And so at first the experimenter, and so there is this button that you can press, and if you press the button, a carefully calibrated pulse of electricity goes down the wire and stimulates the brains under the tip of the electrode for, let's say, 200 or 300 milliseconds. There is a pulse of electricity activating neurons in that area. And two neuroscientists by the name of Olds and Milner discovered, so they were pressing, stimulating the rat, and they discovered they can kind of do conditioning. They can condition the rat in the same way that you can condition a dog by giving it food. They can condition the rat by pressing the button of the electrode For example, if you want the rat to go to the corner of the cage, you press the button when the rat is in the corner, and you don't press it when the rat is somewhere else. And the rat kind of starts going to the corner, which means that this button press is somehow reinforcing. It feels good. So the key insight was to allow the rat to control the electrical stimulation itself. And so you have this very important concept of self-stimulation, and it is very closely related to addiction. This is what addicts are doing when they inject drugs into their veins. They're self-stimulating certain parts of their brains. But as we'll see, these data are just absolutely mind-blowing. Nothing compares to just stimulating electrically the structure directly, not kind of injecting some drug into the veins and kind of waiting for blood circulation to carry it to the brain. So the results show that rats, there are places inside the brain. So there are all kinds of places inside the brain. There are places that the rat presses the button once and then it avoids it at all costs. Just that button is bad. I don't want anything to do with it. But there are these other places in the brain where if you insert the electrode there, the rat goes crazy and they frantically self-stimulate for very high rates, like 2,000 button presses per hour for 24 hours non-stop. Like once every second, press the button like crazy, non-stop, foregoing food, drink, sleep, sex, anything. They just are glued to this pedal. And at some sites, notably this lateral hypothalamus, remember when I told you the hypothalamus, which is in control of the homeostatic regulation of the body and on top of the pituitary gland, which is the master endocrine system. So that same hypothalamus is now the place where if you put an electrode and the rat will self-stimulate nonstop, and there doesn't seem to be a satiation point. There are some other locations in the brain, the rat will stimulate, let's say, 50 times and then go to sleep. But if you insert it in the lateral hypothalamus, it could stimulate itself to physiological exhaustion until it is just physically unable to press the button anymore. Moreover, you can ask the rat how much, ask kind of metaphorically, how much pain are you willing to endure in order to gain access to these pedal? And the rats will brave painful shocks to their feet for an opportunity to self-stimulate, shocks that are a lot stronger than what the rats are prepared to endure in order to find food, even if they are very hungry. You keep a rat hungry for 5 days, and then it still wouldn't cross this grid on the bottom of the cage because the grid is electrified, and it hurts the rat to go over it. But if the rat knows there is this magic button on the other side and I can self-stimulate the lateral hypothalamus, then the rat will cross that. And similarly, drug addicts, human drug addicts, they go to buy drugs and they get beaten and knocked and really abused, and then they still go back the next day because they need their fix. Okay, so I have these vintage videos. Let's see, that's probably the next. Oh, before that, there is a video. I simply don't have a slide about it, but I will play you the video. It's fascinating. But here is this rat brain. This is the spine here on the right side. This is the cerebellum. This is the front of the brain. This is towards the nose of the rat. So this is the rostral direction. This is the caudal direction. This is the bottom of the brain. This is the medial surface. So if you slice the brain sagittally like this and then open it up and look from the middle, so the cortex is around the top here, and then this is the medial forebrain bundle. And so all sites around here are self-stimulating, in the sense that if you implant an electrode in any of these areas, the rat will self-stimulate. But especially so in this location here, which is the lateral hypothalamus. So see, the thalamus is here, the hypothalamus is under the thalamus, and so that would be the hypothalamic site. Now, there are the videos. Let's not forget them because they're fascinating. So share with sound. So let's see if that will work.
Working in Montreal in the 1950s, James Olds devised a crucial test. He worked out a new way to, in effect, ask animals how they felt.
This is a device which trains the animal to turn on the stimulus for himself. Here you notice the animal wandering toward the pedal. When he first touches it, he gets no stimulation. It's not connected yet. Then, one time he touches the pedal, it does turn on the stimulation. You can see the lights go on.
Now, the interesting thing about the old stimulation, the rewarding stimulation, was the strength of it. It was somewhat different than rewarding stimulation for food and water in that there was no satiation. Animals would continue for hours and hours and didn't seem to satiate. They didn't feel full. They would just keep doing it for long, long periods of time. They would do it to exhaustion.
Now, I will electrify the grid. The animal must cross an area that gives a very painful shock to the feet in order to get to the pedal and stimulate the brain.
And a rat wanting brain stimulation would brave a shock stronger than even a starving rat would brave to get food.
This shows a rat is willing to pay a very high price in order to get to the pedal.
Human addicts will pay just as high a price for their rewards.
Okay, so when we talk about motivation and alignment and all of that, that is obviously extremely relevant. Okay, so now neuroscientists since the 1950s have figured out that these kind of medial bundle, this pathway involves the dopamine system of the brain. So there is this neurotransmitter called dopamine, which is very important in reinforcement learning and reward signaling and reward prediction error and so forth and so on. The two major sources of dopamine are two. So one of them is the structure called ventrotegmental area over here, and another is the substantia nigra over here. And so the ventrotegmental dopamine projects along the purple lines, and the substantia nigra dopamine projects along the orange lines. And so stimulating these sites along the pathway interacts with the dopaminergic system. And there are these, overall, there are four major neuromodulatory systems. The dopaminergic is one of them, but there are three others: serotonin, noradrenaline, and acetylcholine. And notice, the sources of all of them are based on the same overall design. The sources are in the brainstem. Low, deep down, the operating system of the brain. So the operating system is this core. It has the motivational circuits. It has all of these things. We're still working. We scientists are still figuring out what is involved in what and all of that. But so there are four different chemicals. Each of them has sources in the brainstem, and then they project to the rest of the brain, and all of them kind of go along this path, kind of horseshoe shape. Okay, let's keep going so I can get at least until the end of the self-simulation business, and we will pick it up next time from there. So positive versus negative valence. Emotion studies, valence is one of the foundational terms, foundational concepts. So positive valence is associated with appetitive behavior. So when the rat goes and presses a pedal, that's appetitive behavior. When it eats food, that's appetitive behavior. When it copulates with a mate, and has sex, that's appetitive behavior. So you do something to get something from the world, that's appetitive behavior. Electrodes in most parts of the midline system, this self-stimulation rates. So this is in the context of these self-stimulation studies with rats dating back from the 1950s. So self-stimulation is a clear form, kind of extreme, exaggerated form of appetitive behavior. And this indicates a rewarding effect. Negative valence, the bad stuff, is associated with aversive behavior, avoidance behavior. So the electrical shock on the floor, the rat doesn't want to step there, it avoids it. In some of the lower parts of the midline system, there was a punishing effect that the animal would press the lever once and never go back. And getting ahead of myself here, similar experiments were done with some patients, human patients, for therapeutic purposes. And so one of them, here, I got ahead of myself, but I'm aiming at this lowermost bullet here. So there was a particular electrode. When the patient pressed it once, said, "Oh, don't do that. That's very bad." It felt sick all over. It just feels, I'm absolutely sick. My whole body hurts. And to make sure this region was not stimulated again, the patient ingeniously modified the hairpin to fit under the button to guarantee that the button cannot be pressed on by accident. So that would be aversive behavior. You tried once and you don't want anything to do with it, then you take active measures to avoid it. And now, these are all behavioristic terms. They don't talk about subjective experiences or psychological concepts. They correspond to the psychological dimension of pleasantness and unpleasantness. There is this mind-body problem, and so it's complicated, but psychologically, positive valence is experienced as a positive, pleasant sensation, and negative valence is experienced as negative, painful, or discomforting, unpleasant sensation. And I have 6 minutes. Let me tell you about, and I hope I can go over time by a couple of minutes. So deep brain stimulation in human patients. There is this fascinating book by Robert Heath, 1996. A lot of the stuff there is kind of borderline unethical. The scientific community is kind of half embarrassed by this guy, so it is not cited in many of the textbooks. But it is available, the copy of the book is available online from this link, and it is absolutely fascinating, and it's also published in journals, peer-reviewed journals, including the most prestigious journal, Science. And so there were these patients with intractable mental health issues of various kinds. So epilepsy, which didn't respond to medical treatment, it didn't respond to electric shocks, didn't respond to anything. And so as a kind of measure of last resort, there were these electrodes implanted in their brains. Similarly with schizophrenic patients, very, very hard cases that have seizures many times a day and so forth. So in those extreme cases, it was decided that it is worth trying implanting electrodes into their brains, into the deep subcortical structures of their brains. This is called deep brain stimulation. It is still performed now routinely as a treatment for Parkinson's disease. The electrodes by the 1970s, so this research was done in the 1960s and 70s, and it could provide both electrical stimulation and also you can squirt drugs through a little tube. The electrons were hollow from the inside and you can inject certain drugs into targeted to circumscribed locations in the brain. And patients would repeatedly self-stimulate some sites and steadfastly avoid other sites. So the stimulation was mostly associated with the septal region, followed by the periaqueductal gray and the lateral amygdala. And now here's when they're humans as opposed to rats, they can actually tell you what they felt. And this is the new thing. We can actually talk to these people and they'll tell you. And so here's one example. Chemical stimulation using the neurotransmitter acetylcholine. So a tiny drop of acetylcholine was injected down the tube into the septal region of a 30-year-old epileptic woman. She became euphoric, often experiencing sexual orgasm. After the first acetylcholine treatment, her seizures stopped and did not come back for 6 months. So the brain is this absolutely mysterious organ. There was a woman, she had multiple seizures every day. They did not respond to pharmacological treatments that you take orally, but if you give it acetylcholine directly to this particular part of their brain, there are no seizures for 6 months. Now, about 24 patients, we have data in the book. Heath reports data from about 24 patients, and several of them reported their experiences. And this paper here, published in the American Journal of Psychiatry, has a few of those. So you can retrieve it if you're interested. So people say things like, it feels great when I press that particular button, stimulating that particular electrode in the particular place in my brain, it feels great. And oftentimes they have sexual thoughts, also elimination of bad thoughts. If they feel bad and you press the button, now they feel good. Sometimes they feel drunk, describe it as happy button, again, elimination of bad thoughts. Now, here's a particularly striking example. One of these patients had these agitated, violent fits, and during one of those, the experimentalist kind of pressed the button without telling the patient. Somebody just gave this electrical stimulus. This was electrical stimulation, not chemical. The first example is chemical. The last example is electrical stimulation to the septal region. By the way, the septal region septum is here near the front. So near the top of the whole chain. So the septal region, during this fit of agitated, violent, psychotic behavior, the stimulus was introduced without the knowledge of the patient. And now, almost instantly, the patient's behavioral state changed from one of disorganization and rage and persecution to one of happiness and mild euphoria. He described the beginning of a sexual motive state. He was unable, when questioned directly, to explain the sudden shift in his feelings and thoughts. Like, I was in rage and now I feel great. And I don't know why. And it correlated precisely in time with the pressing of the button. Quite striking data, as you can see. So bear with me just for this one last slide, and then we'll get to the social anxiety experiments next time. So negative experiences, all of these were positive. Some of them were negative, and the negatives are studied much less for ethical reasons, but there are some cases just by accident, something was stimulated that generated this very bad effect. So here's the most striking example in the book by Robert Heath, available online. So direct quote from the book: the most profound example of a negative phenomenal experience was elicited with stimulation of the mesencephalic tegmentum of patient JM. He became, after the stimulation, he became murderously rageful as he threatened to kill the physician standing next to him. When the stimulus was turned off, the patient was perplexed by his reaction. I have nothing against him. He was just the closest to me. He then remembered similar rageful feelings he had had in the past, calling them, under a term of his own making, gorilla-osis. I felt like a gorilla and wanted to kill. And so in jail, there are people they every once in a while attack the guards out of nowhere. And so the human condition involves cases like that. Now, in some cases, so this one was about rage. And last but not least, there is fear. Fear could be induced in many places in the brain. And so when the electrical stimulation induced fear, it was usually usually described by feelings of discomfort and anxiety unrelated to the immediate environment. So you just feel afraid of nothing in particular. The patient was aware of the profound emotion that mobilized him for swift action to defend against the person or object nearest to him. His behavior was fully conscious and well coordinated, not an automatism or sham. Okay, so I'm out of time. I have to stop kind of in midstream here. What's going on? I pressed escape. So I'm sorry for the cliffhanger. I hope you have found this whole stuff as fascinating as I have, and I will continue talking about it on Wednesday, that is tomorrow. But now I encourage you to take a brief break and then probably come back for Kalina's lecture in 25 minutes.